Good morning, afternoon, evening. My name is Fabi van Cappel, and today I will present my thesis, distinguishing hereditary from sporadic colorectal cancer using unsupervised DNA methylation clustering, a proof of concept for veteran exposure biomarker discovery. Here is a clinical challenge. Lynch syndrome cancers and CpG island methylator phenotype, or CIMP positive sporadic cancers, look identical at bedside. Same symptoms, same presentation. Distinguishing them currently requires expensive genetic testing. But here is real-world impact. Veterans exposed to burn pits who develop colorectal cancer have no objective molecular evidence to support their VA disability claim. They can't prove their cancer came from toxin exposure versus hereditary or random chance. This research aims to change that. My goal was to develop an unsupervised clustering method using only methylation data, no predefined labels. The hypothesis is that methylation patterns alone can differentiate cancer subtypes at a molecular level without providing the algorithm with specific search criteria. The ultimate application, providing objective molecular evidence for toxic exposure claims, evidence that doesn't rely on expensive genetic testing or subjective and presumptive clinical judgment. Quick biological foundation. DNA methylation is an epigenetic modification. It's stable, yet environmentally responsive, making it ideal for detecting exposure effects. Lynch syndrome involves inherited mutation in mismatch repair genes, causing microsatellite instability. CpG island methylator phenotype, or CIMP, involves aberrant methylation silencing the same genes sporadically. Same endpoint, different causes, and methylation pattern should differ. I use TCGA-COAD data, the gold standard for colorectal cancer genomics. I needed 3 datasets: clinical data for outcome, mutation data to identify Lynch syndrome cases as my validation labels, and methylation data for nearly 500,000 CpG sites. The critical step was identifying patients with complete data across all 3 types. That's my analysis cohort. Not all patients had complete data. By intersecting clinical, mutation, and methylation dataset, I identify a multi-omic cohort of 292 patients with all 3 data types available for analysis. The overlapping region labeled multi-omic cohort encompasses patients with all 3 data types available, allowing for comprehensive multi-omic analysis that links genetic, epigenetic, and clinical features. Existing research primarily uses supervised learning because we already know who has Lynch syndrome. But for veterans, without genetic testing, we need unsupervised methods. There is also no established framework using methylation signature as evidence in military disability claims. My thesis addresses both gaps. Data quality is critical. I implemented at least a 7-step pipeline following Illumina 450K best practices, starting with tumor filtering, reducing from 352 to 292 samples. Then I applied aggressive quality filtering. That was removing probes with more than 10% missing value, single nucleotide polymorphism or CNP associated probe, cross-reactive probes, and sex chromosome probes to avoid confounding. Beta mixture quantile or BMIQ normalization corrected the type 1 and type 2 probe biases. I also apply KNRS neighbor or KNN imputation to handle the remaining 0.19% missing value. The result? 10,000 high-quality, high-variance CpG probes selected from nearly 486,000 with 99.8% data completeness. Immediately after the pipeline, I wanted to see how it looks like. I just wanted to see it. So I ran principal component analysis, or PCP, to show the pre-processed data. Even before clustering, you can observe a natural structure emerging here. The color gradient along the principal component 1 suggests meaningful methylation variation. PC1 captures nearly 30% of the variance. This confirmed that the data was ready for clustering. We had structure. Why consensus clustering? 4 key reasons. It's robust to outliers, it determines optimal K objectively, it quantifies cluster stability, and critically, it's unsupervised with no predetermined labels. The core principle, subsample, cluster, track. We construct a consensus matrix that records our sample pairs to cluster together across iterations. High consensus mean biologically stable, meaningful clusters. The implementation process exhibits testing multiple values to find optimal clustering. Consensus clustering was performed for K2 through K10 with 1,000 iterations per run. You can see the output here. Testing K2 and K3, tracking means consensus score. For K3, we achieved 0.97 consensus with cluster size of 106, 73, and 130 patients. These results support biological hypothesis of 3 methylation subtype, CIMPI, sporadic, and Lynch-like. This is robust methodology, 9,000 iteration across K2 to K10 provide objective evidence for cluster selection. The multi-step quality control pipeline and consensus approach overcome limitation of single clustering runs, ensuring reproducible results. This graph shows why selected K3 higher consensus value closer to 1 indicate reliable, reproducible clusters. As K increases, consensus decreases, because finer separations become increasingly complex to reproduce consistently. At K3, we maintain a mean consensus of 0.97, high stability. Going to K4 drops dramatically. The elbow here confirms that 3 is the optimal number of biologically meaningful clusters. So I wanted to try 2 independent validation approaches. On the left, you have principal component analysis, a linear method. Principal component 1 captures 30% of the variance with clear cluster separation along this axis. On the right, you have T-distributed stochastic neighbor embedding, or T-SNE, a non-linear dimensionality reduction method that preserves local neighborhood structure. Both methods independently confirm the 3 distinct methylation subtypes. Notice here, in green, cluster 3 is spread in both plots. This heterogeneity is biological, reflecting the diverse nature of sporadic cancers. This is not a methodological artifact. This heat map shows the top 100 most variable CpG probes across all 292 samples, ordered by cluster. Blue indicates hypomethylation, red indicates hypermethylation. The patterns are striking. Cluster 1 is predominantly blue, global hypomethylation. Cluster 2 is predominantly red, the CIMPI phenotype with widespread hypermethylation. Cluster 3 shows mixed pattern, the heterogeneous sporadic group. This visual confirmation matches exactly what we would expect biologically from the literature. We'll focus on the highly mutated clusters. For instance, you can look at the cluster 2 in Lynch mutation, is about 24%. This is a critical external validation of our unsupervised clustering. Remember, we identify this cluster using only methylation data with no prior knowledge of mutation status. Now we are asking, do these methylation-based clusters correspond to known molecular subtype? The answer is a clear yes. Lynch mutations, cluster 2, shows a highest rate at 24% compared to only 8% in cluster 1. BRAF mutations, this is striking. Cluster 2 has 52% BRAF mutations compared to just 3% in cluster 1 and 7% in cluster 3. BRAF mutations are the hallmark of the CIMP-high pathway. MSI-High, again, cluster 2 dominate at 52%. MSI-High and BRAF together define the serrated CIMP pathway, and our methylation clustering found this without being told. BRAF mutations, here, cluster 3 stand out at 62%. BRAF is associated with a conventional sporadic pathway, exactly what we would expect for a heterogeneous cluster. The key point here is that all 4 markers are statistically significant with p-value ranging from 0.006 to 2.7 times 10 to the negative 9. This is not a chance. Our unsupervised methylation clustering has independently rediscovered non-cancer biology. Beyond molecular markers, do our cluster correlate with clinical outcomes? Age, highly significant at p equals 8.2 times 10 to the negative 5. Cluster 1 patients are the youngest, with a median under 60. Cluster 2 patients are the oldest, with a median around 71. Gender, not significant, balance across clusters. This confirms our clusters aren't confounded by sex. Tumor stage, significant at p equals 0.0065. Cluster 2 has the highest proportion of stage 3, around 45%, meaning more advanced disease at diagnosis. Cluster 1 and 3 show more stage 2, indicating earlier detection. This is clinically consistent. Younger cluster 1 patients are likely to have hereditary predisposition and may benefit from family screening programs. Older cluster 2 patients present later onset with more advanced sporadic disease. So, what does this mean? This study demonstrates that unsupervised DNA methylation clustering can objectively identify colorectal cancer subtypes with distinct etiological origins without requiring expensive genetic testing or predefined labels. The methodology provides a foundation for developing molecular biomarkers to support Veterans Affairs disability claims. If we can distinguish exposure-related cancer from hereditary or sporadic disease using methylation signature, veterans would have objective evidence for their claims. This is a proof of concept. The next step is to validate it in veterans cohort with documented exposure history. So I will close this with a quote of Sam Keen. Genes are like the story, and DNA is a language that the story is written in. To add to the quote, I will say that methylation is how the environment edits that story. Thank you. I'm happy to take your question.